Hi everyone, welcome to our third in our molecular pathology end-to-end testing webinar series. Part 3 is all about analysing and reporting results, and I'm going to do the second part of that about reported results, and the first part of it about data is going to be done by our guest presenter Cathal O'Brien, even from Dublin. Next slide please. So he's going to talk to you about what to do if you've got too much data, and then at the end we're going to have a live question and answer session. Can I ask that you all remain on mute for the duration of the webinar and that you raise any questions via the chat facility on the menu. This webinar will be available online afterwards. So on to Cal, the first slide of his, I think, should be next. There you go, and I shall leave you in his capable hands to talk to you about what to do with the data that you generate in NGS.
Thanks very much, Jenny. So the title today is what to do if I've got too much data, and it's a problem that a lot of us face. Used to be an issue years ago, but as NGS has become more commonplace, it's something that we have to contend with a lot in molecular pathology labs. So we'll go to the next slide there, please. Okay, so there's no pictures on this, but anyway, we'll go with the text. So the big thing about clinical labs is that we're going to get asked very simple questions, generally speaking. From a pathologist or a geneticist point of view, they're really just looking to answer, something simple like, will this patient respond to a particular tyrosine kinase inhibitor? And the answer you might give is yes or no. And if you actually look at it, if you type that into a file and you measure it and you say, oh, well, actually you get the size of it, it's going to be 2 or 3 bytes. It is tiny. So the actual data volume that is required to be sent back, if we're talking about in computation terms, is minuscule. Now we're going to add that to a report format and maybe it gets to a few hundred kilobytes, but really it's still a very small file size. But that's not what we tend to hear when we're talking to people in molecular diagnostics labs, because people talk about data. So if we go to the next slide there, please. Okay, so molecular diagnostics, in particular NGS, is data in issues. So data has been growing steadily in molecular diagnostics labs, but really it's the advent of next-gen sequencing that has really taken, I suppose, resulted in a massive increase in data. So whereas before we could have been generating megabytes, we went to generating gigabytes very quickly, and not too long after that we're generating tens and hundreds of gigabytes at a time. So go to the next slide, please. So I try to think of an analogy sometimes when I think about, trying to explain to people NGS. And again, this is a crowd of people who presumably know how next-gen sequencing works, but I was trying to think of an analogy. And I'm going to go with this one and see how far I get, because I haven't thought it through fully, but we'll see how this one goes. So if someone asked you to try and figure out who was sitting in the wrong seat in a football stadium, but you were very limited in what you could do. Now, if someone said, okay, right, I want to know if the person in seat 26A is the correct person, well, you just go over there and you ask their name and say, you Cathal O'Brien, they say. No, you say, oh, okay, you're in the wrong seat. But if you actually have to check everyone at the same time, and we're looking at something that's a huge data problem, you can say, well, there's only a few things I know. I don't know everyone's name. I don't know where seat they're in. I have 4 things I can measure, and that's just hair color, black, brown, red, or blonde. And that's the only things you can collect. And you can't actually say where those people are sitting. When you gather the data, you can only just say, well, I think this row of people should have 2 people with black hair and 2 with red and then 2 with blonde, and that's how I'll know. If I see that, I think everything will match up. You start getting to like a very obscure way of trying to answer a problem. But really, that's kind of the way NGS works. And if you look at this stadium, maybe there's 50,000 people in there, you could be looking at something, a data problem that's 100 times that. So imagine a stadium is 100 times bigger. You can only measure hair color as your output. It's inaccurate, and you generate this massive file, and you have to try and figure out from that who's in the wrong seat. And that's pretty much what we do with next-gen sequencing. We've only 4 things we can measure, and we're not always the most accurate in those measurements. And then we have to put those into strings, and then we see where we think in the genome those strings belong. So we end up generating a lot of data. When we have figured that out, and we figure out, okay, right, we think there's a change from a C to a T at this particular position, then there's, I suppose, the work of the genetics, or sort of the variant classification begins. But we're not even going to talk about the variant classification, because that's, from a data perspective, fairly small. I'll just mention it a bit at the end, but the classification isn't the big thing. The big thing when we're generating lots of data is at the analytical side of things. So we're generating millions and tens of millions or hundreds of millions of NGS reads, which are about 100 bases long, give or take, depending on what sequencing you use. And we have all of those contained in typically one file. And that's going to be, that file is called a FASTQ file or a BAM file, typically. And that just contains the raw reads of A, C's, G's, and T's. And sometimes those can be aligned and they can tell us where we are in the genome, but generally we're looking at a massive data set relative to the volume of data we'll go down to. So a whole genome sequencing file could be 100 gigabytes. A targeted panel might be a gig. A small targeted panel might be 100 megabytes if you're looking at something really focused. So we're looking at massive jumps as you go into whole genome sequencing or exomes. And all of that data have to be in a format that can be interrogated and interpreted, and you can't give that file back to the clinician, obviously. So you have to get that condensed back down to a reasonable result, and then that has to go into a report, which Jenni will talk about reporting formats after this. So we are talking about answering the clinical question. We generate in the labs, nobody gives us a load of data, we generate a load of data, and we have to, so we get a small data problem, which is a question at the start with the patient's demographics, generate a truckload of data, give a result, and then we're stuck with all this data that's left behind. So if we go to the next slide, please. So the big question then is, what do we do with these large volumes of data? So all the data in the middle, we've answered the question, report's gone, everything's cool, we hope, and we're left with all of these data. And people are often reluctant to discard the data. So the question is, what do we do when we have so much data? Bin it is possibly reasonable in many circumstances. We'll talk maybe about a few sort of suggestions later. But generally speaking, we're always trying to toe the line between keeping the data or discarding it. If it's irrelevant and unnecessary and we'll never use it, we really need to discard it. But generally, there's that thing that's going to be going off in the back of your mind saying, we could use this, or it could save us money, or it could be useful for this patient in the future. And so at a service level, and actually as a service level nationally even, it does need to be considered what data we generate, how long we keep it. So if we go to the next slide, please, and what the purpose sometimes of doing that is. So the path of least resistance, I think for a lot of services, a lot of hospitals, a lot of health systems, is if you have cloud computing set up, you send it to the cloud and you don't really think about it anymore. And since cloud computing has become very popular and popularized a lot of buzz about it, it seems like the sort of thing, just put it in the cloud, it's easy. And it is very easy actually to put in the cloud. Unbelievably so. I mean, we've all shopped on Amazon. I see Jeff Bezos there. He's kind of the person who, his company anyway, Amazon, one of the most profitable arms of his company is cloud computing, Amazon Web Services, for good reason. I mean, it's as easy to use their web service as it is to order things online if you're into computers anyway, and you say, oh, I want to rent a server. It takes about 5 minutes. So cloud computing has meant that it's very easy to do this. So if we go to the next slide, it might work for you, right? So it's relatively inexpensive for long-term storage. And when you look at the cost of, let's say, setting up a server, keeping a sort of a network attached storage or some sort of direct attached storage and having backup and all of those things in place, cloud computing can be relatively inexpensive. And I do have a caveat on that, unless you end up storing a lot more data because you always just send it to the cloud, and that is possible because you start, instead of saving, a terabyte of data, you save 100 because it's just like it's in the cloud. It's, you don't have anyone from IT calling you saying, okay, you need to buy a new hard drive or invest some money here. You don't really have anyone applying the brakes until the bill comes in at the end of the month or the end of the year. And if you can justify the cost, sometimes that will mean that you will store a lot of data. So there is a caveat to that. It's massively scalable. So you can go from generating those small panels, which are 100 megabytes, and you can scale up to 1 gigabyte panels and potentially scale to 100 gigabyte whole exomes. And from a storage point of view, you won't really notice the difference. It'll all get stored. You don't have to invest in anything. You can just take another 100 gigs of storage and you're golden. And you can automatically apply data retention rules. You say anything that is in the cloud for longer than 2 months, I want that to move to cold storage. And anything that's in there longer than 2 years, I want to move to glacial storage, which is really slow storage, but really cheap. So really and when I say you don't need to take any action or put any policies in place, what it means that from a lab, biological and clinical function point of view, you can really say, we'll keep everything and we'll use it when we need it. So you don't really have to make any hard decisions about what files you need to keep, what data, you keep it all. And so potentially that's great because you never have to take any hard decisions. And as long as there's money to cover it and IT are okay with it, you're golden. So go to the next slide, please. So why it might not work. So maybe not Jeff Bezos, and you don't like Jeff Bezos particularly, but you may not like cloud computing. And lots of people do not like cloud computing. They like to see the physical hardware in the lab. They like to hear it, humming away in the corner, heating up the lab, whatever it is. Some people like the physical hardware. And I think there is a degree of predictability to that, because you can buy an instrument, you know what it costs you up front, and it's really only costs you electricity after that, and you have to pay for maintenance, of course, but the cost is very controllable. And so what it means that if you make a capital purchase, say I'm going to buy a server, I'm going to keep it for 5 years, you're actually going to work within the limits of your spend. So your spend is very predictable, and that's not the case with cloud computing. So the other downside is that it can be a waste of resources. And if you overuse your data storage, it becomes relatively expensive because you're storing a lot more data than you would have done if you took that controlled capital expenditure move at the start. The retrieval of dump data, and it's not dump data, it's data that we're just kind of shoving into the cloud, can become problematic, because what will happen is the volumes of data will become significantly larger than they would if you had a discard policy or a do not store policy for some of your data. And where that becomes difficult is if you have to do retrospective analysis. You've got maybe 1,000 files where you would have previously had 100. It's going to take more than 10 times as long to get those data back. If you want to do some sort of analysis, it can really slow down data retrieval. And if you have 10 times more files and you have to do a retrospective analysis of everything you keep on file, it's going to take 10 times longer. So you could end up having to do, if you have to do a retrospective audit for whatever reason, something you suspect something in a pipeline, it might take a year to churn through the data, and it will cost you an absolute fortune. Whereas really you should be saying, okay, well, I'm going to be judicious about this. And if you did that with wet lab analysis, that's what you would do. You would say, I'm going to take a representative section, I'm going to look at that, and I'm going to see how that fares out. So it can become problematic. So what might happen in that case is you have so much data, it's practically useless because it's too big to analyze or too big to do anything with. So again, it's one thing to store it, it's another thing to get it back and do any further analysis on it. Your institute might not work well with cloud solutions. I mean, that's experience we've had here, where our IT department is not, or has not traditionally been comfortable with cloud solutions due to funding and a number of other things. But it means it's very difficult to push cloud services because we don't have the people here to work with that. And I think in the NHS that may not be the case, but in, let's say, isolated institutions like our own, that might be a problem. So it can take a long time to get a cloud solution working as well. And the other part of it is if you're stuck with vendor lock-in, it means cloud solutions and cloud storage may not be an option for you. So if we go to the next slide. So vendor lock-in then is just the concept, and I'm sure most people are familiar with it, of being linked to a particular vendor. You can't get away from their system. And if, for those of you old enough to remember, this is a zip disk. We have zip disks in our department, and we've no zip drive. You can't even buy them for a lot of modern computers. So we have a lot of data stored on these, and it goes into long-term storage, but it's stuck on a disk, on a zip disk. And so if at any point we ever had to go back and look at these files, we don't even have a drive that will read that disk. I'm sure someone will have one somewhere, will charge us a fortune if we have to access it, but we're locked into that vendor's format. We can't, it's not open. So we actually can't, buy a zip disk drive from someone else. In fact, they're quite slow in usage, so most people wouldn't, well, by modern standards, most people wouldn't have one anyway, even if they were available. But we're locked into that vendor's format. And that can also happen with bioinformatics software solutions. So if we go to the next slide. So if you have a pipeline that a vendor has provided for you that you've purchased, you can end up getting locked into that. It's the sort of black box approach that people frown upon, especially people who are into bioinformatics. You just don't really like that black box format. And the reason for that is that everything is locked in. You can't see what's going on under the hood. And if you do look at it, it's not really covered. Solutions like this can be particularly useful if you don't have a lot of in-house bioinformatics support, you're getting started, getting set up. But the problem with it is, and let's say, for example, you buy a server from a vendor, you know, I'm not going to name any particular vendors, but you buy a server from a vendor, or you put it on their cloud solution, and they do all the data processing for you. You're locked into them for, well, forever, really, for as long as you need to get access to that data and be able to retrieve it. That's definitely the case for cloud. You have to do some local backups and things, but if you can't break the data out and look at it in, you know, look at the individual components of everything and store them, you can end up being locked in. So that server needs to stay active for 20 years if you've decided you need access to those files for 20 years, because you've no other way of retrieving the data from your analyses. So servers that typically last 5 years, they're not going to last 20, so you can actually realistically use them in 20 years. So you end up having a very finite timeline on how long you can access that. It means you're locked into the vendor, and you have to pay them legacy support for machines that don't really work, or they have to migrate all that data to a new instance, so that, again, you're locked into what the vendor is supplying. So it can be an issue. It's not the case, well, I suppose it is the case with some solutions, but generally there are ways to kind of work with vendor solutions or work around your own in-house pipelines. Go to the next slide, please. So that you can kind of have the best of both worlds. Someone has set up the pipeline for you, but you can get the open source, open readable data files that you need. So example here is we have our NGS data on the left, and typically that's going to be your FASTQ or BAM file. So these are big data files, like I mentioned, 100 gigabytes could be typical for a whole genome. That data goes into a vendor supplied or in-house bioinformatics pipeline. And again, documenting that is really important, because we need to know what happened to data, how they were processed, what file versions were used, what software versions were used, what database was used at the time. So we do need to document that. So that's all part of our good lab practice and our SOPs and things. So we'll have our version control pipelines, which is the second thing on the bottom row there. And so the pipeline is going to basically look at your raw data file, and it's going to produce a list of variants. This is kind of like your raw VCFs, your variant call format, I think is the term for it. So I've just put down an asterisk there in the bottom right. So VCF or other human readable variant file. Now VCF doesn't really, if you look at a raw VCF, it's not that readable, but you can decipher it. And it has a particular format that is easy to look at. You can look back at it in 20 years because it's only a text file. You can open it in Notepad on a Mac or PC, whatever you prefer, and you can look at the actual raw data and you can figure out what's going on. So you don't need to use a proprietary software package for that. So a VCF is a really good file format for archiving the calls, provided you trust the pipeline at that point. Next thing you're going to do is you're going to filter. So you're going to use a filtering script of some descriptions, and then you're going to have, so you're going to have your filtered VCF, and then you're going to possibly have a variant classification record. So that's deciding whether this variant or this is pathogenic or benign or whatever it might be, and then you're going to issue your report. So along the way, there are certain file types that you're going to keep, and as you move from left to right across that particular slide, the file size decreases dramatically. So the report being the smallest in terms of the actual data, your VCF might be tens of megabytes. Your FASTQ or BAM, by contrast, could be hundreds of gigabytes. So trying to find the right storage solution and what you will store is sometimes, the VCF will be, to my mind, a minimum. But if we go on to the next slide, I'll just kind of talk through a few of the different file formats. But we do need policies, local policies for data retention and discard. And I do think that a lot of the RC path guidelines that we would tend to use here, and I know you use them in the UK as well, a lot of those don't really specify retention times for data, but there are sort of analogous situations. You might save a worksheet, whereas you might save details of a pipeline or something like that. So there are things where you can draw parallels. So go to the next slide there, please. So for nucleic acid, nucleic acid, even though it's not data, it contains the potential to generate a lot of data. You might store that, and again, we're thinking about wet lab situations here, traceability, future reanalysis, retrospective trend analysis, lots of different things you could do with nucleic acid. And I suppose by guideline, we'd keep that for quite a long time anyway. Not optimal for stability, it can go off over time. So if a 20-year-old sample is used, it may not be optimal compared to using one that's just been extracted today. And it is a very cost-effective long-term solution for storing data, because you can reanalyze it in the future. You can analyze it for something else. You can use a different panel. There's lots of different advantages to it. So nucleic acid is pretty much a no-brainer. And then in between that, we have lots of other things that we could look at. So we go to the next slide. And this is, then we get into the data side of things. The FASTQ, there's really only one reason you would keep a FASTQ, is that if you want to reanalyze with a different pipeline in the future. So sometimes people might use a virtual panel. They might only look at a small subset of it, and they want to go back and look at it again. And so you might keep it for as long as it's useful clinically. But it's not optimal for traceability because it's not going to contain any information about the procedure. It's a lot of ACs, Gs, and Ts along with quality scores, and that's really all the FASTQ is. So the pipeline and documentation for your pipeline and who set things up on your worksheets, that's going to be a lot more informative from a traceability and future audit perspective. And it's not optimal for long-term storage because the cost typically of storing large data files. The most cost-effective approach for this is just medium-term storage to permit reanalysis within the expected time frame for patient benefit. And that sometimes that can be difficult to nail down because you think, okay, well, we might have something that we might want to use this file for. But if the file has been analyzed in its entirety, so let's say a small panel would be a good example, you've analyzed it for all potential targets, you haven't like done a bioinformatics subpanel, and you're unlikely to go back to it, the reasons for holding on to the FASTQ start to fall off fairly quickly. But it is, I think it's perfectly reasonable to keep it for a limited amount of time, maybe a year, two, whatever, but it really is probably of the data files, excluding, say, pure raw data, is one of the first ones that really should go when you're considering what you need to cut. Again, it's an opinion, but I'll stand over that one really just on the actual complexity and the utility of the actual file itself and the data it contains. Go to the next slide, please. So the VCFs are arguably probably the most useful. Provided you trust the pipeline is validated, you haven't deviated from anything, you haven't made any modifications to it, it's well documented, you know how the data have been processed. You can use the VCF to go back and look at filtering, you can look for different variants that you didn't report the first time but were called by the bioinformatic pipeline. You can look at variants again in regions that you didn't know were important when you go back to it. It provides a good degree of traceability, small footprint for archival, but it's not optimal for reanalysis. So if you have a new pipeline or a brand new tool that's really shiny and will pick up on low-level variants that your other one didn't, it's probably not going to be the most useful. You're going to need the raw data for that, but it's arguably the case that you're not going to have a lot of situations where that would apply. The most effective long-term or cost-effective approach here is long-term storage, same for worksheets. It doesn't cost a lot to store, and I would store it pretty much as long as I would store the worksheets for the same basis. It could be 20 years in some cases, but that's reasonable because the files contain enough information that you can go back and you can reanalyze within limitations of your pipeline, and you have all those variants and everything is traceable, and it's a perfectly reasonable file, I think, to keep for a long time. And the equivalent of VCF should also be considered as that's what we use for, let's say, fusion calls or something. So the next one then is, go to the next slide. Trying to think, yeah, variant classification records. And these I just mentioned because these are the classification records for you have a variant and then you have to classify it as pathogenic or benign or whatever it might be. And that's something you need to revisit quite a lot. That's pretty much a live record because you're going to, the next time you see it, you're going to say, well, how many cases have we seen? Do we need to do anything? Do we need to update the classification on that? It's not optimal for archival. You don't really want to just say, I'm going to put that in a file and I'm going to send it away to storage. A database where you have live access to those variants, to that is really important, and you need to be able to update it. So your classification records probably need to live in a database that's updated as frequently as needs be and is backed up on a very routine schedule. I mean, I'm just suggesting here once a day, you know, a daily backup, weekly backups, and then immutable quarterlies, but there would be some sort of backup schedule in place for that database. We'll go on to the next slide. So in conclusion, I think the open source, or I suppose file formats you can access using something like Notepad is really important. If you get into a proprietary format, it can be very difficult to get anything back out of that. And VCF is an open file format, and that's kind of the one I would advocate most for as a minimum storage long-term and minimum cost-effective. Storage and discard policy is quite important because it allows you the comfort of saying, look, we've made a clear decision about why we keep these files for as long as we keep them, and we discard them after a certain time, and there's good rationale for that, and it's discussed and hopefully agreed at a departmental level, if not national or at least best practice guideline level as well. And then the data storage and discard policies should match the clinical need. Again, there are certain parallels that can be drawn between, let's say, worksheets or sample retention from the RC Path guidelines as they currently stand to data, but it would be nice to see the RC Path guidelines getting updated to include certain recommendations on data storage as well. So that's me for now, and I think then we'll maybe do some questions later on if there's time as well. Thank you.
Thanks, Cal. That was a really interesting. Data's obviously such a huge part of new technology that people are now using with NGS. But I'm going to go back to what we were talking about in actually reporting the data itself. And I'm going to talk about standardizing quality in reporting molecular pathology data. And I think my slides are on automatic go forward, which is going to be a problem, but we'll hopefully sort it out. So I'm Dr. Jenni Fairley, and I'm one of the Deputy Directors of GenQA, and I look after the molecular pathology EQAs. And we always get a lot of sort of questions about what should be in the molecular pathology report. And there's a lot of things that we need to properly interpret a report. It could just say we found a BRAF mutation. Well, that's not really helpful because there's lots of different BRAF mutations. So you need some kind of nomenclature to describe the mutation. You need to have a reference sequence to tell you how to, can you go back a slide, thanks. You need some kind of reference sequence to interpret that mutation. You need to specify what you've tested, so to work out what you might not have found because of the test you've done, because that can be really important for treating a patient, because it might be that you've done a limited test, but actually the oncologist wants to see a much bigger selection of things that you've tested for. And then some kind of interpretation of results. And this can be slightly controversial in the molecular pathology field, but I think we're coming to a place where it is about interpreting the genetic results in the context of the patient's backgrounds. And it's not really about saying which therapy is suitable for the patient itself, but which therapy has been associated with a particular result. And I'll come back to that in a minute. And then the second part of my talk, I'm going to talk a bit about something that we've also alluded to in the previous part, and that's about classification of variants. And I'm going to talk a bit about our solid tumor somatic and variant interpretation EQA, and also some of the tools that GenQA is trying to put in place to help with that. So next slide. So there are lots of different guidance documents out there about what should be in a molecular pathology report, and actually what should be in a genomic report. And these are generally guidelines, and they have certain suggestions, and they are mainly do come up with the common things within the guidance. And we've recently, GenQA, some of the members of GenQA have been involved in updating the recommendations for reporting of diagnostic genomic testing results. And these can pretty much be applied to molecular pathology, even though the document doesn't specifically mention molecular pathology. So next slide, please. So the principles that apply to reporting, as outlined in that document, are that it should be a clear report, you should be able to identify the patient, it should be concise, accurate, answer the clinical question, and be as fully interpreted as is appropriate. So next slide. And I'm going to talk about how a report can be clear and accurate. And so the next slide, you need to use internationally agreed nomenclature or a concise statement to give a result of a report. And one of the things that we discourage, for the most part, because we do appreciate there are some types of results that do need to be described as positive and negative, but we discourage the use of positive and negative in respect of, have you found a variant or not? You shouldn't be using positive or negative. Where you might use positive and negative are immunohistochemistry results, where the staining, the only way you can really describe it is positive or negative. And the other type of results recently, where I think at the moment we don't have better terms, are when we talk about HRD testing, homologous recombination deficiency testing. Can we go back a slide? And in that case, we have to use positive and negative at the moment because that's the terminology available. On reports, we often have people say to us, oh, but the oncologist only understands V600E. I shouldn't have to use nomenclature to describe it. It's fine, you can write V600E on your report, but you should be using the internationally agreed nomenclature as well. So you can use legacy or historical descriptions, but it should be clear that they're being used as legacy or historical descriptions. You need to include sort of reference ranges and cite the sources. I mean, that's not quite the same for this, but that's part of the ISO standards as well. And in terms of now, we've been talking about doing large panel testing. Now, you could list all the genes you're testing in a large panel, and probably it'd be good to have it some way available, but putting every single gene you test in the main part of the report is probably not appropriate these days, as it probably does distract from the actual clinical results you're reporting. So, I mean, this is a bit of a wide question about how you report the scope of your testing and what you've been testing. But certainly doing it all in the report at the front is probably not the best plan. So next slide. So I'm going to talk about why we need to use nomenclature to report accurate results. I've got a couple of examples that I can show you. So next slide. So this is about, this is some examples of how we had the same fusion transcripts reported in one of our sarcoma, in fact it was the NTRK EQA, I think, on the sarcoma EQA, but it is an NTRK fusion that's found in sarcomas, and laboratories reported it in all these different ways. And if I was a clinician reading all these different reports, I might not realize that they were all the same thing, which in fact they were. And so there is a need to try and use international nomenclature to try and harmonize what's written in reports, so that wherever you get a report from, you can understand the same results. And in this case, I've ticked the one which would represent correct nomenclature according to the current guidance. In addition, you want to really have a clear statement of results, and I've got another example of a report which I feel didn't have a clear statement of results. And next slide. Has it gone? Here we go. So this was a result. Again, this is in a fusion panel result, and this is the whole content of the report that was reported by the participant. So what they've reported was that their fusion overall call was positive, and then they detected all these different things, and then they didn't say anything else. Now, to me, I don't know what that, if I was a clinician reading that, I'll be looking at that going, oh, there's something about ATF and EWSR1, but I don't really understand what any of this means. Are these important transcripts? Is there any implication for the diagnosis? I think you need to clarify results like this and explain a little bit more about them. Reference sequences are also really important for this. And again, I've got some examples of why you need to use a correct reference sequence and why you need to use a version number of the reference sequence. The next slide. So this is again an NTRK, another fusion, and fusions are throwing up some of the most difficulties we've seen with reporting, I have to say. And this is about the different reference sequences have different exon numbering for the fusions. So according to different reference sequences, the fusion would be between different exons. And if you're trying to interpret whether a fusion actually produces an open reading frame or whether there's any stop codons that prevent it opening a reading frame, if you don't have the correct transcript, you wouldn't be able to work that out. So you don't know whether or not it's a valid fusion. And this was one that was particularly complex because there were two reference sequences used for AFAP1, which was one of the partners in the gene, And then for the NTRAT2, there's two different versions of the reference sequence where the exon numbering changed between the two versions. So that's something you have to be quite careful of. If you use the most updated version of the reference sequence, is it really aligning with what you've used in your software to generate the data? So next slide. As I mentioned at the start, specifying what you do for your test is really important, because it needs to be, your report needs to say what you did to get that result, but it should be concise, and it should be enough for another scientist to be able to reproduce the results and for the readers to understand what was done, and importantly, the limitations. So you can include technical sensitivity or practical resolutions, which will be based on internal data and validation testing. And then you would potentially list any kits you use, if they're commercial kits, software packages, and you might include, with the caveat we're talking about at the beginning about having too long lists, details of the regions of genes tested. The final part of this is about answering the clinical question and fully interpreting the report. So next slide. So what you really want to do is interrogate the results you find to provide an overall answer to the clinical question in a clear and concise way. Now, it might be that you could argue that a concise way of saying the results is, well, we interrogated to find out if there was a clinically actionable variant, we found this variant. However, you really need to include a conclusion which is understandable by the different clinical professionals. So you need to say whether that variant is important or not, really. The report should contain up-to-date information that you can interpret the results with. So perhaps literature, reference sequences, obviously, the databases used, maybe disease-specific databases if they're used, and also suggest further tests. It's really important how we report no clinically significant findings. The report should say that you haven't found a clinically significant variant, not normal or negative. You shouldn't really be including benign or likely benign variants, because that will just cause confusion. You need to have stated what has been tested with any limitations. What might the test have missed that we've got a negative result? Because it might be our test has missed, not tested something that represents 10 or 20% of variants in a particular gene, in which case you probably want to do further appropriate investigations. If your test has got incomplete or failed testing, this should be clearly stated on a report. So that, again, the details of what testing you did get results for, what you would recommend for further testing, and if appropriate, requesting a repeat sample to do that testing on. Next slide. So the last part of my talk is going to be about classification of variants. And in the past, we were only testing a limited number of biomarkers for very specific hot spots. In solid tumours, the classification of variants wasn't something that was particularly important. With the introduction of larger and larger gene panels, more clinically relevant biomarkers, and more clinical information about genes, classification of variants has now become a much more hot topic and something that we do need to start to think about in the solid tumour world. Next slide. And there are multiple different classification systems which are in use at the moment. They're for different variant types and different clinical settings, but they're all there to try and standardise the approach. I will say that in solid tumours, there is really, I think, two main sets of guidelines which have been introduced in the past few years, which are starting, well, three sets. The pathogenicity guidelines produced by the ACMG in 2015 are often used to determine whether a variant is pathogenic or not, and that has been applied to the solid tumour setting. Then in 2017, Lee et al. Did produce guidelines about interpreting sequence variants in cancer. This is much more about actionability of variants. In 2020, I think there's a new set of guidelines from Riggs et al. Is about copy number variants. In the UK, the ACGS has got rare disease class variants. So that there's some new ones have just come out. There's also CanVig, which looks at cancer susceptibility genes. And in the UK, we've been working on new versions of somatic variant guidelines to try and encompass the best of all worlds. So next slide, please. And this very much reflects what we've seen in our somatic sequence variant EQA. We've been doing this for actually, I think, 4 years now. We don't have results yet for 2023, and this is 2023. But what we've seen is a large percentage of laboratories are using the AMP-ASCO-CAP guidelines. These are the Lee et al. Guidelines published in 2017 about actionability. A lot of labs will also use these in combination with the ACMG. This is Richards et al. 2015, which is about pathogenicity guidelines. But there are a bunch of other guidelines that people are using, which are available out there. SCAT is a European Society for Medical Oncology version of the AMP-ASCO-CAP guidelines, is fairly similar. And the guidelines that people use will depend on which case what the variant is. That was particularly clear that we sent out variants that were fusion transcripts, and people would use different guidelines from them from what they might use for example a BRCA variant. So the guidelines used will depend on the individual laboratories, but also the cases they're trying to interpret. Next slide. But because there are all these different guidelines, it does mean that there is still differences in the way that laboratories classify variants. And these are even within labs using the same AMP-ASCO-CAP Lee et al. Guidelines about actionability. And looking at the different cases we sent out for the EQA, you can see there was quite a lot of variability in whether they were classified as like clinically actionable tier 1 and tier 2 variants, or non-VUSs and tier 3, or benign variants tier 4. Now we have seen more, certainly in 2022, the benign variant, we definitely saw more harmonization between the laboratories, whereas the tier 2 and tier 3, there is still a lot of variability into what laboratories are calling a VUS or a clinically actionable variant. So next slide, please. We also looked at both the ACMG guidelines, which were pathogenic, determine whether something's pathogenic or not, or the new guidelines that were produced in 2022 from Horrick et al., which were about oncogenicity and are on the similar setup to the pathogenicity guidelines. And again, for the pathogenicity, likely pathogenicity, VUS, likely benign, again, we saw quite a lot of variation in whether something would be called likely pathogenic or a VUS. For example, case 3 in 2021, you can see there's quite a lot of variability there in that would have actually quite clinically significant results, in that if you call something likely pathogenic, it's more likely to be actioned, whereas if it's a VUS, it's much less likely to be actioned. The difference between pathogenic and likely pathogenic is not so strong. So next slide. Variant interpretation is a very complex procedure, and these EQAs that we run are generally laboratory based. However, variant classification is done by individuals with their own knowledge and experience. And we recognise that there's a need for individual assessment and competency checking. And because of that, GenQA has introduced Jenni, the Genomic Online Individual Education Platform. The next slide, please. And we've been working at trying to bring individual education, so sort of for individuals, like to look at the competency that an individual has in aspects of the laboratory medicine. And we've looked at various different areas that at the moment, including variant classification. We've got a couple of EQAs, a couple of Jenni modules, which provide classification modules. One that we run annually about BRCA and HRR gene variants, and that is usually available currently without cost. But we also have an online variant classification EQA. Currently, that's designed for germline variants in a number of diseases, both for single nucleotide, the small indels, and copy number variants. There's a cost associated with this. You can find details on our website. We are investigating doing something similar for somatic variants, but we feel at the moment, because it's still quite variable, that there's still a little way to go before we can perhaps do this for the somatic variants. But we're also looking at other types of individual competencies. We run an EQA looking at tissue assessment from FFPE tissue, which I think we've talked about before, called TissueEye. We've been looking at training and evidence and competency with a suite of genomic videos. And then the last Jenni modules that we've set up relatively recently are about variant nomenclature. So next slide, please. So we have at the moment a variant nomenclature module, which is available as a free trial until the end of April. And these provide online competency assessment for HGVS nomenclature and ISCN. And anybody who uses these to describe genomic variants can test their knowledge using these free modules. You can register on our GenQA website. And if you go to www.genqa.org and follow the links to Jenni, you should be able to register it and use this to look at how you report things using ISCN or HGVS. Next slide, please. I just want to thank everyone within GenQA for all the help we do. We have a huge team of assessors that help us with all our EQAs, and also a large number of experts in our specialist advisory groups. We have to utilise our laboratories for sample sourcing and validation, but mostly we have to thank all our participants, because we wouldn't exist without them, and really do appreciate your participation in our EQAs. So next slide, please. So now I've got, I think, one question that has come in from the panel, and it's about variant interpretation. So I think there's always a question about variant interpretation, about which guidelines should we be using to interpret variants. And this one is a quite interesting one, where it's a UK laboratory, but they're getting samples from overseas. So which guidelines should we be using? One's based from the UK, or one's based from the country where the samples are being sent from? In reality, what we'd like to think eventually is that we would have guidelines that would be universal, that wouldn't be country specific. But we do know that which drugs are approved are country specific. So it's a bit tricky. Cal, have you got any insights into that one, do you think?
Yeah, that's what I try and avoid as a question. I'd agree. I think the pathogenicity shouldn't vary based on the guidelines. The guidelines should be equally accurate in predicting variant pathogenicity or oncogenicity, but there are some things where they're going to differ. And again, my experience of that is limited. I don't work in genetics per se, but we do see that there are some cases where one set of guidelines might move from a VUS to a likely pathogenic or otherwise. And I think those are the cases where something is borderline, that it maybe merits looking at multiple guidelines and just saying, okay, by one guideline it might be this, and by another it might be that. But generally speaking, again, in my limited experience, if something is barn door pathogenic in one set of guidelines, it will be barn door pathogenic on the other.
I think the difficulty is in actionability, and I think that's where we struggle, because the Lee et al. Guidelines about actionability are definitely highlighting whether they're FDA approved therapies and things, and obviously that doesn't apply to all countries. I think the way the guidelines are written just now is confusing. I think it would be better for it to be either FDA approved therapy, or like the evidence is strong enough. It shouldn't just be about approved therapy, because it's like variants that have been found that even if the drug's not available, it's important to note that it doesn't change that the variant may give eligibility to a certain drug trial.
An interesting point, and just something I'd noted down to ask you at the end, because you're talking about some of the guidelines, and some of the guidelines are about how you classify a variant as oncogenic or likely oncogenic. And the second part of that is, now that you've classified it as oncogenic or likely oncogenic, how actionable is it? And so the Lee et al. Guidelines are about actionability, but they make no reference to how you classify the variant. And then you have something like HORAC or the ACMG, which will allow you to actually classify the pathogenicity. So I think there's probably two sets of variables that are required for classification. I mean, what's your take on it?
Yeah, I would agree. I think it's very difficult to be able to classify a variant as actionable without looking at whether it's oncogenic or pathogenic, other than the barn door ones we already know about, where you wouldn't need to classify them because we already know. So it's a really tricky subject area in, I think, solid tumour testing, because the question we're asking a lot of the time is this patient going to respond to a particular drug? And actually, you don't know that unless you try it, because if there's no clinical trials, what evidence do we base it on? It's sort of guesswork, isn't it? So I think it's always it's a difficult one in the area of solid tumours. But I think the idea of having something, well, you say this is an oncogenic variant or likely oncogenic variant, and then you go, well, we don't know, but there's data suggests that variants in this gene with similar levels of oncogenicity might respond to the variant.
And I wonder, is that due to some of the guidelines being a process, say BRCA guidelines? If you're testing, query HBOC and you're testing BRCA 1 or 2, the outcome from that testing is it's either going to be pathogenic or likely pathogenic, and the implications that are very clear. Whereas for the somatic thing, it might be, will a patient respond to therapy, like you said, but the guidelines don't really address that, because you just say it's oncogenic or likely oncogenic. Yeah. And now we suppose that they're going to respond, and we have to do a separate thing to say, well, will this likely oncogenic variant predict response? So we end up kind of doing a double assessment, I think, within the somatic side of things.
I agree. But I think that's perhaps appropriate, because you could argue that the first stage of that is definitely a laboratory-based assessment of oncogenicity, and then the second part of it is perhaps much more appropriate in tumour boards or with consultation with oncologists and that kind of thing. The first part of that is, I think, very lab centred, and the second part of it is much more complex, and where the idea of tumour boards or multidisciplinary teams are going to become more and more to the fore as well, I suspect. And there's an elephant in the room with all these guidelines, is that a lot of the guidelines focus on interpreting individual variants, but we actually know that variants turn up together, and we don't really have good ways of interpreting multiple variants yet in cancer genes, I would say.
And even I think resistance variants are one of those ones that tend to be underrepresented in the likes of COSMIC, because they're only detected in resistance settings. So if you just look and say, oh, there's only 3 reports in COSMIC, it may downplay the performance of that in a treatment resistance setting as well.
I definitely think somatic variant interpretation is a work in progress.
Definitely.
but very interesting one. So next slide, please. So we welcome participant feedback on all our webinars, and those of you who've registered today should get a survey sent to you following the webinar. And this webinar will be available with a link and hopefully we've set it up properly. It'll give you a participation certificate. Our next webinar will be in May, not April, like it says there, but it will be in May, and it will be the last part of our molecular pathology end-to-end testing webinar series. And we've got a couple of presentations on about why you might want to do EQA, and also what's new in the world of molecular pathology. And that's going to be on the 14th of May at 12 noon. And is that the last slide? I think so. Yeah, thank you very much for attending today. The webinar will also be available eventually on YouTube within about a month's time. And we look forward to seeing you at our next set of webinars in May. Thank you very much.
Jenni, good luck.